Initial microscopic examination of semen specimens. Microscopic examinations require standardization to ensure the comparability of interlaboratory data and minimize individual evaluation variables. The World Health Organization, WHO, has created standardized practices that laboratories performing sperm analysis can reference. The microscopic examination includes 1. Presence of agglutination or aggregation. 2. Motility. 3. Vitality, viability. 4. Concentration. 5. Morphology. 6. Concentration of other cells present. A well-mixed sample is essential for accurate testing and to ensure reproducible results. Mixing can be achieved by placing the specimen container on a moving tray during liquefaction in a 37 degrees Celsius incubator. Without an orbital mixer, a basic mixing could be achieved by approximately 15 to 30 seconds of manual swirling. Another method is to gently aspirate it 10 times through a wide-bore pipette. A semen sample should not be mixed in the original container so vigorously that bubbles form. Semen samples should never be mixed with a vortex as this may damage the sperm. Microscopic observations for agglutination and motility are performed using a wet preparation of semen. The wet prep should be prepared following a consistent process that has minimal impact on the movement of the sperm. The wet prep should be made within 30 to 60 minutes after ejaculate collection. The World Health Organization Laboratory Manual for the Examination and Processing of Human Semen, WHO, 6th edition, recommends placing 10 L of well-mixed semen, preferable pre-warmed to 37 degrees Celsius, on a clean glass slide and placing a 22 mm x 22 mm cover slip over the semen sample. The weight of the cover slip spreads the sample, so a number 1/2 weight cover slip is recommended. Initial microscopic examination of a fresh semen sample should be performed at 100x magnification, 10x eyepiece, and a 10x objective. A phase microscope is recommended. Initial 100x examination is used to observe mucus strand formation, sperm aggregation, or sperm agglutination. When no sperm are evident on the low power, 100x examination, the slide should be examined at higher power, 400x, to determine if sufficient numbers are present to warrant further testing. In the former recommendations, if the number of sperm is less than 4 per high power field, HPF, it was clinically acceptable to report the sperm concentration as 2x106/ml with a comment if motile sperm were or were not seen. However, now it is suggested that sperm be evaluated and counted even at very low numbers. An aliquot of the sample can be concentrated by centrifugation, and slides made and stained for documentation or review for unusual findings. Be aware that centrifugation does not harvest all sperm that may be present and may damage those that are present. Thus, observations such as motility and morphology should never be performed on centrifuged samples. The presence of non-spermatozoa cells, such as epithelial cells, derived from the urogenital tract, and round cells, which include leukocytes and spermatids, can be seen on higher magnification. However, identification and differentiation of leukocytes and spermatids should not be attempted on an unstained wet preparation. These cells can be more accurately identified through the use of PAP or right-stained smears. Aggregation of spermatozoa. Nonspecific aggregation of spermatozoa should be recorded but is usually not clinically significant. Aggregations of this type include a motile sperm adhering to each other in a random fashion and motile sperm adhering to mucus strands, epithelial cells, or other debris in the specimen. Nonspecific aggregation is depicted in the image on the right. Initial microscopic examination of semen specimens, continued. Agglutination of spermatozoa. Motile spermatozoa adhering to each other in a specific pattern is known as agglutination. These specific patterns are head-to-head, tail-to-tail, mixed, or tangled. The sperm typically have vigorous motility but are so strongly agglutinated they appear as a shaking clump. Sperm agglutination is graded and reported according to the degree of agglutination as well as the pattern, see tables below. A schematic of tail-to-tail agglutination can be seen in the image on the right. Additional images of sperm agglutination patterns can be found in the WHO Laboratory Manual for the Examination and Processing of Human Semen, 6th edition. This reference is listed on the reference page at the end of this course. Agglutination of motile sperm cells may indicate the presence of anti-sperm antibodies. Confirmation of anti-sperm antibodies requires more specialized testing of the semen specimen. This is typically performed in specialized andrology laboratories rather than the routine clinical laboratory. Immotile sperm aggregated in nonspecific patterns and motile sperm adhering to mucus or debris should not be misinterpreted as agglutination. This nonspecific aggregation should be noted as present, but it is unnecessary to grade or describe the type of aggregation. Grade 1. Degree of agglutination. Isolated. Sperm involvement. Less than 10 spermatozoa/agglutinate, many free spermatozoa. Grade 2. Degree of agglutination. Moderate. Sperm involvement. 10 to 50 spermatozoa/agglutinate, some free spermatozoa. Grade 3. Degree of agglutination. Large. Sperm involvement. Greater than 50 spermatozoa/agglutinate, some free spermatozoa. Grade 4. Degree of agglutination. Gross. Sperm involvement. All spermatozoa agglutinated, agglutinates interconnected. Site of attachment. Attachment grade. A. Description. Head to head. B. Tail to tail, heads are free and move clear of agglutinates. C. Tail tip to tail tip. D. Mixed, clear head to head and tail to tail agglutinations. E. Tangle, heads and tails enmeshed. Heads are not clear of agglutinates as they are in tail to tail agglutinations. Sperm motility. Motile sperm are critical for fertility. To determine the quality and number of motile sperm in a semen sample, it is recommended laboratories follow the standard guidelines found in the current WHO Laboratory Manual for the Examination and Processing of Human Semen, 6th edition *. To ensure that all staff and laboratories are consistent in their interpretation of sperm motility. Motility should be assessed at a consistent time post-collection, preferably at 30 minutes, but at a maximum of 1 hour. Motility should be measured at a consistent temperature. Room temperature, 20 to 24 degrees Celsius, is acceptable but should be monitored. It should be noted on the report if testing is performed outside the acceptable temperature range. Motility can be performed at 37 degrees Celsius, but most laboratories lack a microscope with a heated stage. A standard wet preparation made for microscopic examination is used to assess motility. To prepare the standard wet preparation, place 10 L of well-mixed semen on a clean glass slide and cover with a 22 mm x 22 mm cover slip. This will provide a chamber depth on the slide that allows free movement of the sperm. Variable volumes of the semen sample and sizes of cover slips may be used but require calculation of the resultant chamber depth, the optimum being 20 m. The calculation to determine chamber depth can be found in the WHO Manual, 6th edition. A depth of less than 20 m will restrict the free movement of the sperm, and a depth greater than 20 m will result in a preparation too deep to keep the moving sperm in focus. Using 10 L of sample with a 22 mm x 22 mm cover slip provides a depth of approximately 20.7 m. Sperm motility, continued. Place the slide on the microscope stage and wait a maximum of 60 seconds for the sperm to stop drifting. A phase microscope is preferred but not mandatory. Examine enough fields at 200x or 400x to enable viewing of a minimum of 200 different sperm in a minimum of 5 fields. Systematically move from field to make sure the same field is not counted twice and avoid areas near the edge of the cover slip. Only intact sperm, defined as having a head and a tail, should be included in the motility assessment. Typically, the progressive forms are identified first, then the non-progressive, and finally the immotile forms. When counting in this sequential fashion, it may be necessary to examine more than 200 sperm. For example, if only the progressive forms have been counted in a field when a total of 200 sperm has been reached, counting will have to continue to include the non-progressive and amodal forms in the field. Failure to do so will skew the result in favor of progressively modal forms. The total number of sperm counted would have to be taken into account when calculating the percentage of sperm in each of the three categories. With experience, it is possible to score all categories simultaneously, thus avoiding the necessity to continue counting beyond the 200 number. An eyepiece reticle is recommended to define the area to be viewed. Samples with very low sperm concentration, cryptozoospermia, may have too few sperm to assess motility. In these cases, the semen should not be centrifuged to concentrate the sperm, as centrifugation can negatively impact motility. The report should include a comment indicating there are too few sperm to perform a motility assessment and an indication of whether any of the sperm seen are motile. Grade of motility. Rapidly progressive, greater than or equal to 25 micrometers per second. Description. Spermatozoa moving actively, linearly, or in large circles, covering a distance from start to end point of at least 25 micrometers, or 1/2 tail length, in 1 second. Slowly progressive, 5 to less than 25 micrometers per second. Spermatozoa moving actively, linearly, or in large circles, covering a distance from start to end point of 5 to less than 25 micrometers, or at least 1 head length to less than 1/2 tail length in 1 second. Non-progressive, less than 5 micrometers per second. All other patterns of active tail movements with an absence of progression, less than 5 micrometers in 1 second, example, swimming in small circles. Amodal. No active tail movements. Replication and verification of sperm motility assessment. Motility should always be evaluated in duplicate. A second 10 L aliquot of well-mixed semen should be placed on a clean glass slide and covered with a 22 mm x 22 mm cover slip. A minimum of 200 sperm should be counted in at least 5 fields and assessed for motility as with the first sample. The results from each of the 2 slides should be compared for acceptable reproducibility by comparing the percentages of the most frequently reported motility grade. The steps are as follows. Determine which motility grade was most frequently reported, the dominant group, then, determine the difference between the percentages of the duplicates for that grade. The acceptability of the difference is determined from the acceptable difference table, from WHO 6th edition, shown below. The table is based on a 95% confidence interval. Some laboratories use a more easily applied plus or 10% limit. If results fall outside the acceptable difference, the counts are discarded due to possible miscounting, pipetting errors, or samples not being mixed properly. If results are within or equal to the acceptable difference, then the average for each motility result is acceptable and can be reported. Note that the percentages should be reported to the nearest whole number. If the sum of the percentages adds up to 99 or 101, then the dominant group is adjusted to make 100%. Example. Motility estimates for 200 spermatozoa replicate counts are as follows. Motility grade. Rapidly progressive percent. Slide 122. Slide 219. Slowly progressive percent. Side 1. 14. Side 2. 9. Non-progressive. Percent. Side 1. 4. Side 2. 6. Amodal percent. Side 1. 60. Side 2. 66. The average percentages for each class are, rapidly progressive equals 21%, slowly progressive equals 12%, non-progressive equals 5%, amodal equals 63%. Note that results are rounded to the nearest whole number. The most common class, in this case, is amodal. The average percentage between the 2 slides is 63%, and the difference is 6. Using the acceptable difference table below, note that at 63%, the acceptable difference is less than or equal to 9, attributable to chance alone. The acceptable difference is less than 9, so the results are acceptable and would be reported as rapidly progressive 21%, slowly progressive 12%, non-progressive 5%, and amodal 62%. Note that because the averages of the first two percentages were rounded up, the sum of the percentages was 101. Thus, the dominant group, amodal, was adjusted to 62%, so the percentages would add up to 100%. Acceptable difference table. Progressive motility. The World Health Organization's current guidelines expanded the previous classification of progressive motility to include rapidly progressive and slowly progressive. Non-progressive motility. The 6th edition of the WHO Manual for the Examination and Processing of Human Semen describes the following as non-progressive motility: absence of progressing, displacing the head less than 5 micrometers in 1 second, i.e., swimming in small circles. Amodal spermatozoa. The 6th edition of the WHO Examination and Processing of Human Semen defines immotility as no movement of sperm head or tail. Sperm viability. Sperm viability, also known as vitality, should be determined if a low percentage of motile sperm are observed. Non-motile sperm may either be viable, live, sperm that are non-motile due to structural abnormalities preventing motility, or previously motile sperm that are now non-viable, dead. Sperm viability should be performed on all semen samples that exhibit less than 40% motile forms. Staining the sperm with an eosin-nigrosin stain mixture is the recommended method for determining viability. Equal portions of well-mixed semen and eosin-nigrosin stain are added to a small test tube, mixed, and allowed to stand for 30 seconds. Traditional wedge smears are then prepared from the mixture and allowed to air dry. This method utilizes the fact that non-viable, dead, sperm have damaged plasma membranes. The damaged membranes will allow the eosin stain to enter the sperm cells, staining them pink or red. Viable sperm will remain unstained and appear white because the eosin cannot penetrate the intact plasma membrane. The nigrosin allows for contrast and fixing for storage of stained slides. The slide is examined at high magnification, 500x or 1000x, and the percentages of viable, white or unstained, and non-viable, pink-stained, sperm are determined. Spermatozoa with stains limited to the neck region are considered viable, live. As with motility, viability should be performed in duplicate, and 200 sperm should be evaluated on each preparation. The average results of the percent viable forms determined on each of the duplicate preparations. The viability stain and slide preparation should be performed as soon as it is determined the motility is less than 40%. The image on the right is an eosin-nigrosin stained slide showing one viable, white, and one non-viable, pink, sperm. Quiz. Lisa, a newly hired and trained medical laboratory scientist, stained a semen sample with an eosin-nigrosin stain to observe for sperm viability. Upon viewing, she noted that the majority of the sperm were stained pink. What is the correct overall interpretation of this observation? Please select the single best answer. 1 sperm are non-viable or dead. 2 sperm are viable or alive. 3 slide is inconclusive as the wrong stain was used. For stain color is not observed for viability, instead the sperm morphology is observed for viability. The correct answer. Sperm that are stained pink are non-viable or dead as they have damaged plasma membranes that allow the eosin dye into the sperm to stain them. Sperm found to be white/unstained are considered viable or alive as they have intact plasma membranes that won't allow the eosin dye to penetrate. Eosin-nigrosin is the correct stain to assess sperm viability. The stain color is viewed for viability. Which tests are recommended if a low percent motility is noted? Please select the single best answer. 1 sperm concentration. 2 sperm viability. 3 sperm aggregation. For sperm motility. The correct answer is sperm viability. Sperm viability is performed to confirm whether decreased motility is a function of non-viable sperm cells or defects in morphology that impact the motility of the sperm. Sperm concentration does not directly impact sperm motility. Sperm aggregation is nonspecific adherence of spermatozoa to debris, mucus strands, and other non-sperm cells and is not related to whether spermatozoa are actively motile or amotile. If a large percentage of amotile viable spermatozoa are observed, this could be indicative of flagellar defects and would be confirmed by sperm morphology testing.